Can invisible marks on DNA help us understand how pollution affects organisms, and even predict the long-term effects of contaminants? That's what the EPIBOOST project set out to discover.
So environmental epigenetics sounds like a real complex word, but let's take a closer look at DNA. Imagine DNA as a cookbook. It's a collection of recipes, but each of us may use different recipes at a different time. And the use of that recipe and when we use what is what we call epigenetics. So epigenetics is really about the use of our DNA and our genes and how we change that. Now, the important thing is that factors like pollution and climate change may also change epigenetics. So they change how we use those recipes without changing the DNA, but with potential significant effects on our life. And EPIBOOST specifically focus on how aquatic organisms, fish, algae, invertebrates, are affected by pollution and how pollution changes how they use their cookbook, their recipe.
The project had one main goal: strengthen the research capacity of University of Aveiro, a leader in ecological risk assessment, by building new expertise in epigenomics, with the support of CSIC and University of Gent, two internationally recognized institutions in this field.
For the University of Aveiro, it has been great because it was a way of capacitating also our infrastructure and our students to work in the field. It is a challenging field, environmental epigenetics, also from the technical point of view, because we need to adjust the information that we get with information already existent on the genomes of the organisms, and we have to tune our protocols for data analysis, for example, to better understand and to reliably understand how epigenetic marks can influence the life history of these organisms and the phenotypes, basically, of these organisms. Also, it was a great opportunity to render specifically our team the skills to manage this kind of larger projects at European level. So there is all these dimensions of capacitation that University of Aveiro gained by working and by collaborating with the advanced partners.
I think EPIBOOST has been a very good chance for students to work with cutting-edge epigenetic techniques, but also to work with practical things like animal welfare, good experimental practices, and how to properly design experiments. That is essential to epigenetics, where even small environmental differences can change the results. The skills they developed were quite wide, thanks to workshops, exchanges, and mentoring. And all the students develop other important skills that can be the communication and critical thinking and even teamwork. The real impact can be seen in some students that are now working abroad, and they are using the techniques they learned, both in epigenetics and also in animal welfare, in other countries.
Beyond the research, EPIBOOST has trained a new generation of scientists, giving them hands-on experience with cutting-edge techniques and international collaboration.
Yeah, I think overall the experience in EPIBOOST was great and allowed me to meet a lot of researchers in this field and learn a lot on environmental epigenetics and bioinformatics, that are two core scientific areas for my PhD. I gained a lot of experience that allowed me to grow as an early stage researcher and hopefully continue working in environmental epigenetics and carrying on the work that I started doing in my PhD and in the EPIBOOST project as well.
So EPIBOOST gave me the necessary tools to help in my work with European CBAS for my master thesis, and in also both in wet lab techniques and data analysis. It also put me in contact with many people from my field area of toxicology, and I believe that all of these techniques and all of this training will prove useful in my future career, and I'm really grateful for the opportunity.
The EPIBOOST project influenced my career future plans. So during this 2.5 years of the EPIBOOST project that I was involved in as a postdoc, I was involved a lot in the bioinformatic data analysis, and this extensive work that I spent doing this bioinformatic, it showed me really how much really I like to do bioinformatic. So yeah, this period working in EPIBOOST have increased my skills in my CVs, and thanks to that I could obtain a scholarship in France to do 1 year postdoc and to work more in epigenetic and bioinformatic.
EPIBOOST really shaped where I am today, since I joined the project as a senior postdoc, and I am now leading the Marine Epigenome Dynamics Lab. All the opportunities and connections I got during the EPIBOOST project opened the door to apply for a Marie Curie doctoral network that opened also like the opportunity to interact with other European research labs, and also this project gave me the opportunity to teach PhD courses and to organize and run the Chromatin Explorer summer school, where I teach dry and wet lab techniques. And this helped to pass the know-how to the next generation of these environmental toxicology researchers.
EPIBOOST helped uncover how contaminants leave epigenetic marks in algae, microcrustaceans, and fish, changes that could one day help us predict the impact of pollution before symptoms even appear.
In EPIBOOST, we mainly work with microalgae, microcrustacean, and fish, representing different levels of marine and freshwater ecosystems.
From a scientific perspective, the most promising results of EPIBOOST is that we really saw impacts of pollutants like cadmium, which is a heavy metal, on different types of aquatic organisms, such as algae, fish, and invertebrates in both freshwater and marine environments. And we saw that these effects are mainly mediated through epigenetics, and that they're really causing significant effects on the health of the organisms. So that's really important for us to know and to help environmental legislation improve so that we keep maintaining healthy aquatic ecosystems.
So we put copepods on different temperature treatments over 5 generations to study the effects of climate change by measuring life history parameters such as reproduction, but also fatty acid profiles and global DNA methylation. We were trying to find out whether epigenetic changes are altering the health of our copepods. This resulted in a different nutritional quality of our copepods, and this is an important finding because they serve as food for fish. This also means that the epigenetic changes we saw are helping with acclimation and might lead to adaptation to climate change.
My work in environmental epigenetics has been an incredible scientific experience because it helped me to understand the specific mechanisms through which toxic substance can influence reproduction, health, and behavior. I plan to keep working in epigenetics, focusing on the regulatory frameworks and ethical challenges, studying the cause-effect mechanisms between the epigenetic modification and environmental stressors, to use all this knowledge and translate it into environmental health regulations.
These findings contribute to building a critical mass of knowledge in the EU, helping bring environmental epigenetics closer to regulatory frameworks for environmental protection.
Environmental epigenetics and its insertion on ecological risk assessment from the regulatory point of view might be a bit difficult at this point, but we trust that in the future it will be possible, and especially it will be important to make these efforts, particularly because epigenetic marks are more stable than other genomic and gene expression marks, and they can be used actually to predict effects in organisms at the phenotypic level. And this would be a great add-on to risk assessment procedures to early understand potential effects in the future. And so we need to keep researching, keep investigating the processes and also the outcomes of the change in epigenetic marks. And as we understand this a little bit better, it will be much easier to incorporate this kind of indicators in ecological risk assessment procedures at the regulatory level.
EPIBOOST may be ending, but its impact continues through new skills, strong partnerships, and a shared commitment to science that protects ecosystems and public health.
I think the main contribution is that we now have a validated suite of techniques and tools for different types of epigenetic technologies available for what we call non-model organisms. Think about copepods, think about specific algae. Usually a lot of epigenetic research focuses on mouse, on zebrafish or on C. elegans, and now we've shown that it's possible to do that also with much more aquatic organisms, adding to the knowledge of protecting all organisms in the environment.
The main contribution of the EPIBOOST project, I believe it was the gathering of a community interested in environmental epigenetics in Europe and also abroad. So, I mean, the networking that our activities allowed, I think they assisted the building of this critical mass to now develop the field at a much higher pace and at a much effective way.
EPIBOOST has helped to build a stronger and more connected European community in environmental epigenetics. By bringing together complementary expertise and training of young researchers, the project has helped building support for future research. We hope that EPIBOOST brings together epigenetic approaches with environmental risk assessment in Europe.